Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. And this is a special episode because we are recording from the Cell Symposium in Exercise Metabolism, Lisbon, Portugal. And I'm super excited because I have today one of the co-organizers, Amber Mueller. Amber, welcome to this podcast, and I'm super excited to hear about everything that you have to talk about cell metabolism.
Thank you so much. I'm thrilled to be here. I'm thrilled to be able to organize this conference and then also sit down and catch up with you.
Yeah, but one disclaimer before we begin. Whatever our discussion is between Amber and I, it's Amber as a person and as a scientist. She's not representing Cell Metabolism, Cell Press, or her employer, Elsevier.
Correct.
Yeah.
My opinions are my own.
Yes. I always begin with this, particularly people like you who were born and brought up in this country, you have a lot of different options. Why science? Why biology?
It's a hard question to answer because, of course, I'm looking back through a lens, right? Of course, I can kind of create a linear pathway to where I am now. But honestly, as a kid, I was just really enthusiastic about animals and nature and aspects, like different aspects of science. I was the kid, I didn't watch a ton of cartoons, partially because I wasn't allowed, but also I was just so much more interested in watching like a National Geographic or, those types of things. I can also say that my dad is a firefighter.
Oh.
And during his training, he had to do paramedic training for a while and work on the medic. And I just thought that was the coolest. He would come home and tell us about these grisly things that he witnessed or saw, and I was just enamored with that aspect. And so, yeah, I at one point I wanted to be an astronaut. I think a lot of kids do. And so I really just went into school, and throughout high school, when you really start to nail down what you want to eventually study in college, I knew that it was going to be science. And then a lot of people were like, oh, you love science, you should go to med school. So I did like the pre-med major at University of Maryland College Park, and decided that I didn't really want to do medicine per se, but I was really interested in the storytelling, the research involved in, getting something from, nothing or from the bench to all the way into the clinic. So I was more interested in that aspect.
No, actually, it's interesting that you mentioned that your dad is a firefighter.
Yeah.
And, a few years ago, I was talking to a few fire families.
I remember. Great work.
No, actually, they're kids. And then I was talking to these kids, and then this little kid who is, at that time, I think he was 7 or something. He said, my dad is a superhero. I said, why? No, when there is a fire and everybody is running away from the fire, my dad goes into the fire and he saves people.
Exactly.
And that's really cool. That really stuck with me. And, those four or 5 days, I interviewed many fire families. I got a very different view of how fire families are and how the kids are, how they grow up. And I guess this storytelling aspect might have come from your dad telling you stories about his firefighting.
I've never actually thought about that, but yeah, it's quite possible.
Yeah. So after undergrad, then you decided to pursue PhD?
Pursue PhD. I did a little stint as a technician in a lab, and it is the lab that I ended up doing my PhD work in. I was kind of told that at the time that I was going through PhD that you kind of had to study a disease. And so in order to get funding or for this reason or that reason. And so I really enjoyed the lab that I was getting experience in as a technician. And I had the opportunity to work on a project that was kind of unrelated to what I was working on as a technician. And so it was a really exciting time in the lab, and I felt really confident and strong mentorship. And so that's why I ended up studying muscular dystrophy, specifically facioscapulohumeral muscular dystrophy.
So what is that? Mouthful.
It's a little bit of a mouthful. It's unlike some of the other muscular dystrophies that result from the lack of structural proteins or proteins that are responsible for membrane stability. FSHD is...
So the muscular dystrophy means how early people can diagnose it. What is the disease? Like in kids and babies, what happens?
Yeah, I mean, it depends. It depends on the type. I think the type that's most famous, people are most familiar with, is Duchenne muscular dystrophy. And that one is much more common in boys because of the basis of the genetics. And
so is it a sex chromosome linked? Or?
Yes, and you can start seeing oftentimes symptoms as early as like toddlers when they basically start to walk. Now they can, but they do it a little bit differently. The way they get up and down is a little bit different. They use their hands more to support their legs, and you can start to kind of notice that early on. That's not the muscular dystrophy that I studied, though. So FSHD is completely different, and it results from basically a truncation at the end of human chromosome 4. And what that truncation does is it makes the region more accessible to transcription machinery. So genes that are not normally expressed in muscle become expressed, and that is like very toxic and very detrimental to the muscle.
So the typical the other muscular dystrophy that you said, Duchenne muscular dystrophy, that's a different disease. That's where the muscle protein is actually not made correctly.
Correct.
But here, other proteins are made in the muscle, and that makes it toxic.
Yes, as far as we understand, yes. And so and interestingly enough, the mouse genome is not structured similarly, at least in this region.
Yeah.
And so it takes a lot of effort to kind of engineer mouse models of this disease. So that was my PhD project, was kind of creating and validating this model.
So that's great. So you got experience in mouse genetics and
epigenetics.
And also muscle biology.
Yes. So interestingly, what makes that region especially interesting is when it's truncated in the form with FSHD, there is hypomethylation of that region, of the genes in that region, and then it makes them also more likely to be expressed. And there are some forms of FSHD that don't have the truncation, but just have overt hypomethylation.
So the DNA is less methylated, which is a sign for
accessibility,
better gene expression, and then the genes are turned on, and then they make toxic protein. In this case. So, when people talk about muscles, there are two aspects, as you said, muscular dystrophy that affects young toddlers and young adults. On the other spectrum, all the adults and people who are getting older, they worry about losing muscle mass. Are there similarity, differences, or can you work on muscular dystrophy and learn something about muscle loss? Because for general public, it becomes difficult. They want to figure out.
Yeah.
They want to know what is the connection.
You can absolutely draw similarities, and there is some overlap in the field, particularly in the stem cell biology field. There are a lot of muscle stem cell researchers that are involved in both muscular dystrophy projects and age-related muscle wasting. But the muscular dystrophies are pretty awful genetic diseases, and so the magnitude, I think, is a big difference.
And affects all muscle types too.
Most of them, yeah. I mean, some of them affect more muscle than others. Some spare the heart, some don't. The diaphragm is in question, depending on the muscular dystrophy. But so there is like some overlap, at least as far as the research is concerned. But there's not as much, I would say, mechanistic
similarities,
right? So sarcopenia, which we think of as age-related muscle wasting, has much more, I would say, an influence of the immune system and of neuromuscular junction deformities or denervation of the muscles. So there are a lot of other factors. Also, sarcopenia is a kind of broad term that describes the muscle wasting, but it doesn't encapsulate all of the various causes, right? So you can have sarcopenia for a myriad of reasons.
Yeah. No, it's kind of interesting that you mentioned one thing that you have to study a disease to get funding. So it's kind of interesting that most of the stuff that we know in biomedical research or what we know about biology mostly comes from broken systems, right?
Yep.
So in some cases disease, and in other cases you make a genetic model where you break a gene and figure out what's wrong with the mouse, what's wrong with a Drosophila fly, or even for humans, as you said, because there was a disease, we went back and learned about what this gene does, or what the chromosome does, or what how the disease develops. So as you studied more and more on this muscle, then what motivated you to have a much broader view and then get into scientific publishing?
Yeah, so I was the person at the bench that, doing experiments over and over and over again. I listened to podcasts a lot. And so it's kind of a full circle moment for me. And I started really getting exciting about aging research. I think I've always been, I would say, fearful of aging, potentially pathologically. I'm always worried about not having enough time.
Yeah.
And it did really, it was kind of a moment that the aging field started to explode in a really positive way, in my opinion. And so I started to listen to these podcasts and, then go down the research rabbit hole, right? Because I was a scientist at the same time. And I kind of, from that work, my horizons opened. And I realized, I didn't have to study one particular pathway and one particular genetic disease in order to, advance in my scientific career. So I went on to do a postdoc. I did aging research, which I loved, and I found to be just really exciting, really thrilling. And of course, there's this huge component of metabolism.
Yeah.
And so as a postdoc, I definitely struggled with work-life balance. I think a lot of people do. I think things are getting better now, a little bit. At least there's more awareness, but it was a big struggle for me. And of course, at the same time, I have this competing fear of not having enough time, right? And I'm spending all my time studying aging, and then feel like I'm wasting the prime of my, youth.
Yeah.
So I loved being in the lab, and I loved the work that I was doing, but I started to think of what else I could do. I noticed in myself that I was the person that would go to conferences and I'd have so much fun talking to everyone else about their work. And then it came time for me to present, and I'd be like, okay, so this is my project, and this is what it is. And it was good. I mean, objectively, it was good work, solid.
So let's put you on the spot now. So what did you work on as postdoc on aging research?
So, right, we can back up. So I was looking at muscle-related epigenetic changes through the aging process using mouse models.
Yeah.
And so I haven't published on that, unfortunately, but I also worked on several other projects in the lab. So we worked on biological clocks a little bit. We had a neuroscience project looking at frailty in mice with a colleague of mine. So,
no, it's, so what you mentioned is really interesting because, in the lab, particularly when you're on the wet bench, if you're curious, just like people who are curious, just like you are,
right.
They would work on 4 or 5 different projects.
Yeah.
They're just curious. You are like in a candy store, and you want to try all kinds of candies.
Exactly.
And then, although you focus on one project that you're forced to publish on that, or your PI wants that, but all those experiences of just dabbling in other projects actually expands your horizon too, because you see what are the tough things on the wet lab. You can ask a jillion different questions, but what you can answer now,
Right.
that becomes more clear, or maybe you learn the challenges when you're dabbling with many things. So did that help you?
It absolutely helped me. I mean, I would go to the journal club, like that was, the whole department, and I'd come back with like 5 collaborations, and my mentor wanted to smack me because, it was like, okay, focus on your own stuff. And then of course I had the wonderful experience of getting scooped, probably because I was spending too much time on everyone else's work. So
But I guess in science, you lose many and win few.
exactly.
Yeah.
And I don't, it was sort of a confluence of factors that led me to decide to apply for the scientific editor position at Cell Metabolism. I think I absolutely could have stayed in the lab and been very happy and successful and gone on to do great things within academia. But it just so happened to work out that I got this amazing opportunity to do the thing that I really loved, which was learn and talk about everyone's amazing research in this field of metabolism, which also has this long thread throughout my research career, right?
Yeah.
Something I was familiar with quite a bit.
So your interest in aging kind of led you to metabolism.
Yep.
So what are the biggest, in your view, what are the biggest breakthroughs or big questions that have been solved or addressed in aging research?
Ooh, it's a big one.
It's a big one, but it's just your personal opinion. I'm not
sure.
Yeah.
Yeah. I mean, therapeutically,
Yeah.
I think these GLP-1 receptor agonist drugs, like dual agonists, maybe even a triagonist here and there, those are really, really exciting on the clinical side of things. And I think we understand pretty well how effective they are in type 2 diabetes and in obesity, but the power of these drugs to solve other potential diseases or manifestations of illness in people is, people are just tapping into it, right? So fertility, for example, is one of the ones that I've heard some work on recently. Even potentially addiction is kind of on the forefront. And so I think that's really exciting. It's one of those things where you have a really interesting drug and then you get to like nail down all of the other myriad of things that it could potentially do.
But if you look at like GLP-1 receptor agonists, initially they were used for type 2 diabetes.
Correct.
Semaglutide and lira. And then obesity. But if you look at many health complications of obesity and diabetes, all age-related health complications, right?
All chronic diseases are age-related, right?
They're also related to obesity, and diabetes exacerbates those age-related disease. So then the drugs that will hit diabetes and obesity will likely have pleiotropic effect, because if we now dial back, GLP-1 receptor agonist essentially makes people less hungry. Let's begin with that. That's kind of the initial thing that people experience, and then they tend to eat less. So in my view, of course, it's a personal view, it's a pharmacological form of fasting. Those who tried and could not fast or reduce their caloric intake, this is a pharmacological way of inducing caloric restriction, at least in the beginning.
Right. Lots of other things we've learned that they do.
Lots of other things, but this is one thing. And then if you dial back and then ask, is there any other drug that does similar stuff, at least pharmacologically induces a biochemical sense of fasting, then milder fasting, then that would be metformin. And as we know now, metformin has been promoted as a drug to slow down age-related disease of aging. So along that line, I think GLP-1 receptor agonists are just, I don't want to use the word metformin on steroids. I mean, the mode of action is very different, and and we're still learning the modes of action of all these drugs. But I guess it's kind of interesting that you in terms of aging research, you latched onto GLP-1 because it does affect many age-related disease.
Yeah, I mean, I think it has, the time will tell, but I think it has the potential to at least extend median lifespan. I don't know if we're going to be extending maximal lifespan in most people, because it's more of avoiding those chronic metabolism related diseases to kind of get you to old age. And then, I'm not sure how much additional length we'll get out of them, but I think that's what most people want, right? Like, I always say, like, I want to die young as late as possible. I heard someone else say that. I didn't make up that expression, but, and I kind of think about that a lot. So.
But I guess this also brings up it reminded me another comment I heard in a conference in a big pharma. I was kind of giving a talk, and then there were some pharma people there. Then it was an aging conference. And then this person said, well, our company has made the biggest breakthrough in aging research by by extending lifespan. And I was shocked. I said, what is that now? Statins. Because if you think about statins, means it's true that the number of deaths from heart disease has gone down. So it's an indirect way to delay death or reduce the burden, comorbidity of heart disease, and also reduce death from heart disease that has extended lifespan to some extent. So in some ways, by treating diseases, we can reduce the burden of disease and add some extra years of life. So I guess in that sense, GLP-1, statins, metformin, all of them, and even antihypertensive drugs, they're all adding years of lives.
That's what we hope. And there's controversy around, some of the drugs that we've mentioned, but yeah, I mean, that's the hope, and we kind of have to see how things play out in humans. I think there's this also controversy around lean muscle mass or lean mass loss with GLP-1 receptor agonists. And some people think it's a big deal. Some people say it's no worse. In fact, it's less than, what you would get from diet alone. Which you do whenever you lose large amounts of weight, you will lose lean body mass. And so it's a little bit of a controversy, but it's also this open area for pharma to then develop, skeletal muscle rejuvenating or at least preventing skeletal muscle loss, which is also great as someone who loves muscle, right? And can have a potential impact even beyond just people that are on, GLP-1 receptor agonists, but maybe people that are experiencing frailty or sarcopenia in late life. So,
but in metabolism, metabolism is is much broader than muscle.
Oh, absolutely.
Yeah. So what are the other areas of metabolism that excites you, or you see that there's big things happening?
Yeah, I mean, it's almost shorter to say the things that aren't metabolism. But I think for me, the most exciting papers are the ones that drop on my desk and I've never even thought of the question before. So that's my little caveat. But the areas that I'm really interested in, I'm very curious about the way that stress interacts with different bodily systems. So we recently published a paper on psychological stress and its impact on the gut microbiome and how that impacts colitis. And then there was another paper recently published from the same group where they showed an opposite directionality. So from gut to psychological stress. So I think, but I'm interested in all types of stressors. So nutritional stressors, but also physical, how different stressors impact the immune system and its ability to, regenerate after injury or induce cancer susceptibility, those types of things. Kind of these multi-system approaches. I think we've done a really good job of studying individual systems and in very specific contexts. And with the amount of data that we're able to collect these days and analyze, with AI, not even having to use our brains for that aspect, I think there's a huge potential for understanding how these different features kind of connect, which is the most exciting.
No, I guess stress, that's a very exciting part too, because one of my colleagues, Sue Keck, who just got elected to the National Academy, she had a beautiful paper on how stress affects the immune system. And when I saw the paper, just like you said, I had never thought about the question.
Oh, yeah.
And then it's really exciting to see this cross organ or cross tissue communication and how that is playing out. And in fact, the GLP-1, the thing that we started with, GLP-1, is a clear example of how a hormone derived from specific types of cells in the gut has so much impact on the pancreas and then on the brain and so many different aspects. So I guess if we look back, most of the important things in our body are done by these messengers that carry messages from one organ or one brain center to the other. All the hormones, same thing.
Exactly.
All the cytokines, inflammatory mediators, they do the same thing. And it's just beginning to see more and more of these hormones or hormone-like molecules.
Yeah, I mean, and coming from all parts of the body, right? Especially if we're talking about metabolites and the way that we just haven't had the tools to identify all of them and identify all of their receptors, right? And I think we're getting close for sure.
So one is, so you mentioned one is the stress and inter-organ communication. What are the other things in metabolism?
I mean, circadian rhythm is incredibly interesting and has the potential to rewrite half of the physiology that that is in our textbooks, right? Just by nature of sampling at the right time and controlling conditions in the appropriate way. So the impact of the circadian rhythm, I mean, you can pick any pathway and probably find a, circadian regulation role. So that's really exciting. I think I'm really interested in sleep. I guess it's also a stressor. I mean, of course I come all from my own, right? My own interests. But sleep or sleep fragmentation or what we call social jet lag. So here, those types of they are stimuli, right?
Yeah.
And I think our bodies are really good at dealing with and adapting to stimuli that happen acutely, but it's the chronic stimulation in these different ways, whether it's sleep fragmentation or psychological stress, nutritional stressors. I'm also pretty stressed about microplastics at the moment. That's more of a toxicology angle, but if someone finds a way that it intervenes in a metabolic pathway, I'd be pretty interested in reading that paper for sure.
Endocrine Society has a lot of position statements on these endocrine disruptors because a lot of these microplastics, they disrupt our endocrine system mostly.
Right.
Or at least that's the mode of action. They go and bind to different hormone receptors. Those are supposed to be activated by our bodily hormones, but now they're activated by these plastic and microplastics.
Right. And what is the actual tangible physiologic outcome, right? And how does that depend on the person or the animal that it's in? Because there's so much heterogeneity. It's kind of we can say in a dish, right? In cell culture, this has this function, but what is the actual physiological function? And then how do we intervene? So all of the topics that I'm talking about that are exciting. It's interesting to figure out what's happening, but it's more important, I think, and equally exciting to figure out how to intervene, right? How to make us more adaptable to these chronic stimuli.
Yeah, no, actually, for microplastic, the other day I was thinking, okay, what would be the right experiment? Because everything in the lab that we use is plastics. So then how can I control? How can I do an experiment where I have non-plastic versus plastic? Come up with a... so it's even doing those experiments was mind-boggling.
Washing glassware, we're back to the 80s.
Yes. I used to wash glassware.
I didn't, thankfully. But yeah.
Now, coming back to sleep again, we are beginning to see some of the results from very severe sleep deprivation, like 3 or 4 days of no sleep.
Yeah.
But most people, we actually don't experience that 3 or 4 continuous days of no sleep, but still that gives us some idea where the things might be moving. But at the same time, I also have some interesting caveat about sleep disruption. So, for example, almost every new mom
Sure.
goes through sleep fragmentation. Means no new mom will claim that they have slept 6 continuous hours for at least the first 6 weeks.
Yeah.
But at the same time, they're kind of resilient.
Yeah.
So then I'm thinking that maybe mild sleep disruption might be very different from chronic sleep disruption or complete sleep deprivation. So
Right.
those will be really exciting to address, to see what happens with just a little bit of sleep disruption and then a little bit more. And why do you break the system?
Right.
Yeah.
And I think some moms would probably say that it's not great. Like it still doesn't feel great. And if there are ways to kind of mitigate the consequences, even if this is like a set time period and not like a chronic lifelong fragmentation pattern, it still gets us, I think, further into kind of, getting the best out of our lives.
I agree that we have to address that because when I was working with firefighters that time, Emily Manogian, who is the lead author, she was actually expecting. And then she had the baby. And then she said, firefighters and new moms are very similar. The only difference is firefighters get days off. And new moms never get days off. So, so it'll be interesting to see what is the impact of sleep disruption. And again, at different stages of life. Like sleep disruption or sleep fragmentation in young adults, teenagers, or college students may be very different from sleep disruption or sleep fragmentation on mid-50-year-old like me. So
Yeah, so it's funny that you bring this up. I have kind of a funny story. My grandmother, who's now passed, when I was starting to get into aging research, she and I would have conversations because I'd learn all of this stuff about gerontology and talk about it with her. She was very influential in my life as well. And I would tell her, oh no, after 65, you sleep for shorter increments. And that's part of the aging process. And she was like, well, I know old people and my friends get good sleep. And it was just so funny that she was so adamant that no, like that's not, I don't accept that that's just what happens when I'm over this age. I'm going to do my best to get good sleep. And if I'm not, like that's a problem. It's not just an inevitability, right? So my family and I, oftentimes, if I say something about some facet of aging research, they'll be like, well, I know old people.
Actually, this is true. Last week, I was talking to someone in London, and this person has been extremely successful in life, in business. And then she said, for all of my life, I was sleeping maybe 4 to 5 hours, 6 hours max. And she had a big business. And now she's not that much into business. And then she said, few years ago, she actually experienced how it feels like having a good night's sleep. And then now she said, I'm addicted to getting 7 hours of sleep.
Good for her.
And she's doing all kinds of stuff, all tricks, not drugs, to get 7 hours of sleep. And she says that this is transformative as she's aging. She's feeling like she's aging healthily. So this is, again, going back to healthy aging. We see those 9 or 10 or 12 hallmarks of aging, but I still have to see sleep.
Well, I think, sleep could probably impact most of the hallmarks, right? It's also part of my work-life balance that I'm really focusing on as well is sleep. People say things like, you can't outrun a bad diet. I would also argue that, you can't TRE bad sleep, or you can't, outsleep chronic stress, right? So all of these facets I'm working to balance in my own life.
No, I guess that's, you mentioned outrun bad diet. So it's essentially all these three: diet, physical activity, and sleep. Those three are
and stress. Which, they're all interconnected.
They're all interconnected. So now, in cell metabolism, as you are looking at many different things. So, very quick question. As we are walking in, you are talking about how you are reading some manuscript or review last night or something. How many papers or manuscripts do you read in a year?
Oh, in a year?
In a week?
So in a week, probably around 14. Because it averages like 2 a day, and that doesn't mean that I do, and that's over 7 days, right? Sometimes less, sometimes, it depends. It honestly, there are like ebbs and flows of paper submissions. So sometimes I go a couple days without reading a paper, and then I read 5 or 6. So the journal gets about 1,500 submissions a year, and there are 4.5 editors.
Yeah.
So you can kind of do the math.
But outside this, these are the new submission, but you are also reading other stuff.
Oh my God.
Yeah.
Yeah. I didn't even think about that. I was just thinking about new submissions.
No.
And I mean, I'm not reading every paper in depth. I'm not like going into detail about the methods unless I have a methodological question. So honestly, it's incalculable.
Yeah.
I couldn't even say.
Because once I was talking to Peter Stern, I guess, from Science. And Peter told me that he reads somewhere between 800 to 1,000 papers in a year. Of course, not, as you said, he's not reading all of them thoroughly, but at least he's getting the gist out of.
Oh yeah, I mean,
1,000.
Every morning I scan bioarchive. So like I'm at least reading the abstracts of 10, 15 papers. I mean, that's just for like what's hot, right?
Yeah.
So yeah, I mean, a huge amount of information that is kind of computing. Some of it sticks, some of it goes on to live somewhere else.
Now, this is interesting because when I talk to my lab PhD students and postdocs, I always tell them that if you read at least 2 papers a day, just scan through 2 papers a day, that would be huge because a lot of people, actually now that we get most of our information from social media and other stuff, a lot of people are actually not reading manuscripts. And that itself is a huge deal.
It was, when I was a postdoc, again, going back into like the kind of things that I would rather do than do bench work. I remember saying to like one of my colleagues, I would love to just take like a paper vacation, right? And just go on vacation, but only read papers, like set up somewhere nice and just take a week and just read everything, because I just felt like I didn't have enough time or I wasn't getting enough. I didn't have enough time to be able to explore kind of curiously. It was like, I have to find this thing and I have to connect it to this. And if it doesn't connect, I don't have time for it. So yeah, and I mean now my whole life is a paper vacation.
No, I like that term, paper vacation.
Yeah.
Actually, sometimes I wish I could take paper vacation, but
exactly.
Like yesterday.
You could be an editor.
Yesterday I was connecting in Munich for 6 hours. I had a 6 hours connection. And that was awesome because I went to this lounge and then I had my caffeine and my water, everything. And then I found a nice desk. I must have scanned through 20 papers and I felt great, like for a long time. This uninterrupted 6 hours. So out of all of this reading and other stuff, now, what are the major trends you are seeing in metabolism research? And I can kind of tell you, kind of prime you in terms of technology. What are the biggest technologies out there that are emerging that will have huge impact on metabolism?
So cost is going down for all of these technologies, even old ones, right, that have been around for a while. So that's hugely exciting. But really the breakthrough is generative AI. And of the moment, just trying to harness the power of generative AI to answer scientific questions or to give us hypotheses worthy of testing, right? And I think we're not quite there, but there are a lot of people and companies working on tools because we are collecting massive amounts of data. I would say the last 10 years has been like the atlas, the decade of atlases, right?
Yeah.
In addition to just like single cell or, transcription factors, there's metabolomics, there's, a myriad of different ways that we're collecting data behaviorally.
Yeah.
And so being able to analyze that and to kind of generate research questions is really exciting.
Yeah. Is there something actively going on that you're aware of in scientific field? Like you said that, can AI generate hypotheses?
Yeah.
And that's a big thing. And have you seen anything going on in any specific, I won't say specific lab because then you'll
Yeah,
find out. But in
yeah, there are absolutely very cool tools that are identifying ligands, for example, that could be really influential for different processes that we didn't know of before.
Yeah.
So yes,
Okay.
it's happening.
So AI, and then what are the other things
I mean,
in technology?
in technology, I think wearable devices on like the clinical or just, therapeutic side that's, not necessarily disease treating, but maybe human optimization in such a way. I think that data is really interesting. And it also, we're starting to get at this ability to individualize at least some interventions. And I think we're going to move more towards personalizing medicine when it comes to drugs. So yeah, those are also exciting areas.
Yeah, I guess continuous glucose monitor is a great example where you can almost personalize nutrition based on CG.
Why stop at glucose? I mean, I think we can start monitoring our immune system, right? Our immune milieu, if you will.
Or stressors.
Our stressors. Oh man, that would be so easy.
Yeah.
I'm sure someone's actually doing that. I haven't seen it, but I'm sure.
Yeah.
So all of these types of things, and really kind of getting at people's own rhythms, their own. Oh, the other big thing is the microbiome, right? And how someone's individual microbiome might make certain drugs more available or less available. And the same with nutrients, the same with kind of any type of lifestyle intervention as well.
No, actually, microbiome, it just reminded me. I read an article a few days ago. There are some very high-end toilets coming to market.
Oh, yeah.
Yeah, those are beyond my budget.
Love it. It's horrifying, but it's amazing.
10 to 20K. But the thing is, every day it just does your poop analysis and urine analysis and will tell you.
It's amazing.
Yeah.
I wish I had one.
Yeah.
I would install it in a heartbeat.
Yeah. I was thinking even simple stuff like, the toilet tissue. That can give us just like litmus paper. The toilet tissue should be able to give us a lot of information. If you have
like a pH strip.
Yeah, like lot. I mean, pH strip is very simple. Then on top of that, you can quote many different things. And I don't know if you are a techie and you have a lot of money, please come up with a better toilet paper that I can wipe and see what is wrong and what is good with me. And I don't have to buy that expensive toilet.
That's true.
Yeah. So in terms of metabolites, my biggest challenge is this. Like if you look at genome sequencing, we can sequence the hell out of it. Like the new sequencers means in every 18 months, the sequencers throughput is doubling. But in terms of metabolites, it's not doubling at that pace. Means we're still below 1,000 metabolites from plasma sample. And I think that's where if we see a big breakthrough where we can go up to say 10,000 and the cost is reasonable, then just imagine in a day, like when I'm doing clinical trials, I have to send patients to do various tests. They are taking almost 5, 6, 7 tubes of blood. I'm like, why?
Yeah.
Means if we can take a oral swab and get the genome out of everybody, then why do we need
or like breath analysis is a cool one.
So those are the things. I think that's where I hope. Again, if Elon Musk or somebody is looking at it, please go ahead and get a machine that will actually analyze every hormone. For example, thyroid hormone, T3, T4, you cannot get from any metabolomics analysis right now. From proteomics, peptidomics, you cannot get even cytokines.
Right.
So that's where I think I wish there are some breakthrough that we can do.
I think
Very few there.
I think we're on the horizon.
Yeah.
I don't think you'll have to wait too long.
Yeah.
Yeah.
Then being at Elsevier, do you, I mean, a few years ago, NIH had a big issue that it mandated that we should do sex as a biological variable.
Yep.
Do you see that is reflecting in publications now? People are addressing both sexes?
More and more, absolutely. I think we could do better. I would say the biggest thing that we can do is honestly have reviewers mention it in every paper, right? Even if it doesn't, even if there's not the budget to redo this entire experiment in male and female mice or whatever it may be, I think just saying it over and over again, it really does come to the forefront of our minds. I mean, that's something that I'm always looking for.
Yeah.
Usually, at least if they're not doing both sexes, they're giving us some kind of reasoning behind it.
Yeah.
But yeah, I think genetic diversity is another big one. And yeah, studying both sexes. It's incredibly important. And people say it's hard, but it's not that hard.
No, it's not that.
People do it all the time.
Yeah.
There was this long-held idea that, oh, well, the female cycle makes things more variable.
No, actually not true,
No.
because even in circadian rhythm, what we are finding is actually female mice have much more robust circadian rhythm than males. When you put high-fat diet, the male mice will, the high-fat diet will disrupt circadian rhythm in males, but not in females. Females are resilient, and it's kind of interesting to see. And then since I'm doing both mouse work and dabbling with human work, the interesting trend is in mouse work, we tend to do everything in male mice historically. But then when you go to lifestyle intervention in humans, women are more likely to enroll in
We love a trial.
interventions. Yeah. So you take the results from male mice, and then in clinic, you're faced with mostly women who are eager to participate. So that's why it's becoming more and more important that we do all this basic science research in female mice and female organisms, and also genetic diversity, because that's a huge thing that is still not addressed in basic biomedical research.
No. And it's hard because you want to control all the factors, genetics being one of them. But at the same time, it means that the context you're studying is so small that the likelihood that it translates into humans is incremental.
It is very small. So that's why, 12 years ago when Cell Metabolism published our first TRF paper, time-restricted feeding, and I would go to conferences, and I was still skeptical that, well, it's only B6 male mice. How it will be translatable? People used to ask me that, should humans do it? I would say, well, if you have a pet B6 mouse, maybe. Fortunately, it reproduced to many extent, but that's true. Like we are humans, we're heterogeneous. Nearly each one of us carries almost 10,000 genes in heterozygous condition, 2 different alleles. And between any two, even brothers and sisters don't carry the same combination, whereas we do all of our research in mice, they have the same allele.
Yeah, I mean, people are making a lot of headway in that regard. So I don't want to discredit the field by any means. I mean,
no, I'm not saying we should not discredit. The point is, there are actually some talks in this conference where people will talk about genetic diversity and how different strains of mice respond to X, Y, and Z. So we are making progress in that case. Yeah. And then the question is metabolism through lifespan. We are seeing a lot of metabolism research in young mice, young adults. But actually there is very little research on metabolism during pregnancy, during fetal growth, during early childhood. Do you see any trend there?
I'm starting to see a trend. I think it's still just a small proportion of researchers that are studying those questions. I think they're doing great work, but it doesn't, it's sort of like, oh, well, if you're studying liver metabolism, you're studying it in, adult male mice. But then if you're studying pregnancy, like, oh, well, that's like a very specific, that's like not, that's for this very niche area. And I think we do kind of have to kind of widen our scope and how we, I guess, interpret those papers and bring them into kind of mainstream metabolism, because it still does feel like a niche field. I also think that metabolism in aging, talk about heterogeneity. I mean, we know in mice and people and pretty much every organism that phenotypes become more heterogeneous with age. And so it makes studying those questions harder, because they're, you have to do a lot more mice to get a significant difference, or a lot more people in your trial to actually see a biological significant trend. So yeah, I mean, we do have to start paying attention to heterogeneity, and somehow like controlled heterogeneity perhaps is the way to go, so that you can still resolve some of those questions at a really granular level, and find those differences.
No, I mean, it's interesting because at one end of the spectrum, we're talking about how to make basic discoveries using very homogeneous, similar mice. At the other extreme of the end, we are talking about personalized medicine.
Right.
Which is, any treatment is very unique to you, and which I completely agree, because almost every medication that we take, the dosing has to be adjusted, and nutrition, exercise, everything is personalized. So I guess this will be a challenge. And
I mean, longitudinal studies is important, within the same creature or human, is one way to resolve.
So what's next for you, Albert?
For me?
Yeah.
Oh, I'm excited to go back to the office and read some more exciting papers. I'm thrilled about this meeting. And just keep learning and keep doing my permanent paper vacation.
No, actually, I will take that term, paper vacation, and I'll tell my trainees that they should take a paper vacation once in a while. And it's really inspiring to talk to you, because on many different levels, you're young, you're so excited about science in general and metabolism, and you have such a broad view, and you're always expanding that view. And thank you for sharing your thoughts on metabolism, your personal journey through life. And hopefully we'll sit down again in
I would love that.
Cell Symposium.
I never thought that I would be in this chair. It's been a really great experience, so I'd love to do it again. Thanks.
Thank you. Thanks a lot.